In our last video of carbohydrates to cholesterol metabolism, we have discussed of cholesterol transport from liver to peripheral tissues. Now let's get our concepts clear on the transport that takes place to and from the liver and learn how actually this process takes place. The liver is organized into collections of cells known as lobules. Each lobule receives blood from two sources. Like other organs, it receives oxygenated blood via the hepatic artery. However, it also receives the venous blood that drains from the gut. The liver is unique in having an afferent venous supply, namely via the hepatic portal vein. This vein carries blood to the liver from the intestines, spleen, pancreas, and gallbladder. This vein transports many products of digestion, such as glucose from the gut to the liver. Important thing to note is that chylomicrons are not transported via the portal vein. They proceed via the lymphatic system before entering the thoracic duct and joining the bloodstream. Well, the products of liver metabolism leave by two routes. Most products leave by the hepatic vein, which is in the center of a liver lobule. However, certain products, such as the bile salts, are excreted via the bile ducts. Until now, we have understood the transport to and from the liver. Now, let's see how cholesterol is being synthesized and transported. Cholesterol, together with other lipids, is transported in blood plasma within large particles known as lipoproteins. Lipoproteins are spherical structures with a hydrophilic exterior and a hydrophobic, lipid-containing core. Their function is to transport lipids in the hydrophilic environment of the blood. The outer surface of lipoproteins is rich in phospholipids and apolipoproteins, which confer upon the lipoproteins many of their specific properties. Lipid proteins are classified on the basis of their density. Because lipids are lighter than proteins, particles that contain more lipids are larger in size but have a lower density. While different types of lipoproteins have different sets of proteins on their surface. These proteins serve as address tags, determining the destination and hence function of each lipoprotein. Before moving ahead, let's discuss these lipoproteins and understand their role in cholesterol transport. We will discuss chylomicrons, a very low-density lipoprotein or VLDL, intermediate density lipoprotein or IDL, low-density lipoprotein or LDL, and lastly, high-density lipoprotein or HDL. If we start with chylomicrons, then it has apolipoprotein, A1, B48, C2, and E. It is present in intestine. It transports dietary triacylglycerol and cholesterol from the intestines to the periphery. Important thing to remember here that it has high concentration of TAG is 90%, whereas very low concentration of protein, which is 1%. Next, VLDL consists of B100, E, and C2. It is mainly found in liver. Its main function is to forward transport of endogenous triacylglycerol and cholesterol from liver to periphery. Level of TAG is relatively less in VLDL. It is 65%. Next is IDL. It has B100 and E. It is basically derived from VLDL. It is used as a precursor of LDL. TAG is 30%. Next, we have LDL. It has B100 and as you can see, it is smaller in size as well. It is derived from VLDL and IDL. It's mainly used for cholesterol transport. TAG is 10%. Lastly, we have HDL. It consists of A1, E, and C2. It is found in liver and intestine. Well, it has very less concentration of triacylglycerol. That is 2%, but has a good number of proteins. That is 50%. Its main functions include reverse transport of cholesterol from periphery to the liver. It stores apoprotein, C2, and apoprotein E, which it supplies to chylomicrons and VLDL. It scavenges and recycles apolipoproteins, released from chylomicrons and VLDL, following lipoprotein lipase activity in the capillaries. At the end, here is a summary for you. You can easily compare lipoproteins in respect to their origins and functions. Now, as you have the basic understanding of these lipoproteins, let's move ahead and learn how it helps in cholesterol transport. As you know, cholesterol is synthesized from glucose by the liver. But some of the cholesterol is esterified with fatty acids in a reaction catalyzed by acyl-CoA, cholesterol acyl transferase or ACAT, to form cholesterol ester. This is hydrophobic, and with its hydrophobic associate, the triacylglycerols, is stored in the core of the nascent VLDL particles. The nascent VLDL leave the liver via the hepatic vein and progress to the periphery. VLDL are secreted directly into the blood by the liver as nascent VLDL particles containing ApoB100. As VLDL pass through the circulation, triacylglycerol is degraded by lipoprotein lipase, causing the VLDL to decrease in size and become denser. In the peripheral capillaries, lipoprotein lipase removes much of the triacylglycerol content by hydrolyzing them to fatty acids and glycerol, leaving the remnant of the VLDL, known as an intermediate density lipoprotein or IDL, which is relatively rich in cholesterol. Well, the cholesterol produced can be cleared from the body by conversion to bile salts, which are excreted from the liver via the bile duct into the intestine. A substantial proportion of the bile salts is reabsorbed and recirculated via the liver in the enterohepatic circulation. Until now, we have understood the concept of lipoproteins and their role in transport of cholesterol in the body. Now, let's move ahead and learn the biosynthesis of triacylglycerol in liver. In addition to de novo lipogenesis, fatty acids are also supplied from adipose tissue, or as dietary fatty acids in chylomicron remnants. The fatty acids are then esterified to form triacylglycerol. The newly formed TAG must not be allowed to accumulate in the liver. The hydrophobic globules of fat must be transported in the aqueous environment of the blood. This is done by enveloping them with a hydrophilic coat of phospholipids and protein to form nascent very low-density lipoproteins or VLDL. The VLDL leave the liver via the hepatic vein and are transported to the periphery. That's how triacylglycerols are being synthesized and transported. Now we will learn the disposal of TAG in the target tissues. The nascent VLDL, while en route to the target tissues, become mature VLDLs after receiving from high-density lipoproteins or HDL, the apolipoproteins, ApoC2 and ApoE. In the capillaries of the target tissues, the apolipoproteins ApoB100 and ApoE bind to the VLDL receptor, and C2 activates lipoprotein lipase or LPL, which is further stimulated by insulin. Lipoprotein lipase or LPL hydrolyzes the TAG contained in the VLDL, producing fatty acids and glycerol. Their fate depends on the target tissue. In adipose tissue, the fatty acids are reesterified with glycerol, reforming TAG for storage. Next, in muscle, the fatty acids can be used for energy metabolism. Next, in various tissues, the fatty acids and glycerol are synthesized to phospholipids for incorporation into cell membranes. While lipoprotein lipase, in the capillaries of peripheral tissues, acts on VLDL to form intermediate density lipoproteins, which are metabolized to low-density lipoproteins. In the liver, ApoB100 of LDL binds to the LDL receptors. These are internalized, and the LDL are degraded to fatty acids, glycerol, amino acids, and cholesterol within the cell. Now, at the end of this, let's discuss disorders of LDL and VLDL metabolism. Disorders of LDL and VLDL includes type 2 hyperlipidemia, type 3 hyperlipidemia, and type 4 hyperlipidemia. Disorder of LDL metabolism includes type 2 hyperlipidemia, whereas disorders of VLDL include type 3 and type 4 hyperlipidemia. Let's begin with the disorder of low-density lipoproteins, or LDL metabolism. LDL has the highest cholesterol content and is the major carrier of cholesterol in the blood. High level of LDL in the blood are associated with cholesterol plaque buildup and cardiovascular diseases. This is why LDL is known as bad cholesterol. Now we will discuss type 2 hyperlipidemia. Patients with familial hypercholesterolemia have very high serum cholesterol concentrations. They die at a young age from ischemic heart disease if they are not treated. The disorder is due to failure to produce functional LDL receptors. The deficit of LDL receptors results in a failure to clear LDL from the blood. The LDL accumulate and cause atherosclerosis. Next, we will discuss the disorders of very low-density lipoprotein, or VLDL. It includes type 3 hyperlipidemia, type 4 hyperlipidemia. Type 3 hyperlipidemia is also called as remnant removal disease. Patients have yellow streaks in the palmar creases of their hand, which is pathognomic of type 3 hyperlipidemia. This is a rare, autosomal recessive condition caused by the production of abnormal ApoE molecules. Since functional ApoE is needed to bind the remnants of VLDL and chylomicrons to the receptor for catabolism, the remnant particles of IDL accumulate. Laboratory tests reveal broad beta band on electrophoresis. Next is type 4 hyperlipidemia. This is an autosomal dominant dyslipidemia characterized by overproduction of triacylglycerols and consequently VLDL. Serum cholesterol concentrations are normal or slightly raised. So now it brings towards the end of this topic. For more videos like these, keep watching sqadia.com.